Okay, welcome to everyone. Good afternoon. Um, so thank you for coming along to the second in this three-part webinar series on the fundamentals of fluorescence microscopy. If you haven't come across me before, my name is Paul McMillan. I'm the manager of the Biological Optical Microscopy platform here at the University of Melbourne. Before we get too far into the presentation, I just want to acknowledge the traditional owners of the land on which we're meeting, which for me is the Wurundjeri Wurrung people of the Kulin Nation here in Melbourne. Pay my respects to their elders past, present, and emerging, and any First Nations people that join us in this meeting today. These webinar series are being run in conjunction with Light Microscopy Australia. If you haven't heard of that before, that's a special interest group of the Australian Microscopy and Microanalysis Society. We have about 250 members from across Australia. If you were to become a member, then you are eligible to apply for travel bursaries for conferences and workshops and lab visits. We do quite a lot of data analysis workshops across Australia during the year as well, and have a regular email which goes around advertising kind of optical or light microscopy related workshops or resources that might be available to people, as well as an annual image competition, which is open to anyone who's based in Australia to submit to, and that will open up later in the year, probably around August, September time. The next major event that's coming up from Light Microscopy Australia is our National Symposium, which will be in Sydney at the start of February next year. That will be 3 days of scientific talks using optical microscopy techniques, with bookended at either end of the symposium with hands-on workshops. And the content that's covered in that symposium will also incorporate bioimage analysis and also intravital microscopy as well. If you want more information, then the website is there. Otherwise, you can follow us on X or LinkedIn as well to find out about any activities that are coming up. Okay, let's go back to the webinar series. So this, as I say, is the second of 3 lectures that we'll be giving. Lecture 1 was last week, so if you've missed that, then I will put up a link at the end which can take you back to a recording. That at this moment will be last year's recording, but I will update all of the recordings for this year once we've completed the webinar series. But you can also download the slides from there as well if you want. So lecture 1 covered fluorescence and fluorescence microscopy. Today we're going to focus on sample preparation, spend quite a bit of time on different experimental applications that you can do using fluorescence microscopy, and cover a little bit on image ethics. And next week, my colleague Ellie Cho will be giving the Introduction to Bioimage Analysis webinar. Okay, so today's talk will cover, as I say, some general advice on sample preparation. This is probably far too specific for individual projects to really get into too much depth and detail, but we'll give some kind of higher level advice on sample preparation. As I say, we'll then spend the majority of the time going through experimental applications, and we'll focus primarily on those that are available at the microscopy platform here at the University of Melbourne. I realize that's not all applications that are available, but that will certainly give you a fairly decent understanding of the majority. And then finally, we'll cover a little bit on image ethics, what you can do and what not to do if you're once you've got your actual data, and then that will kind of segue in hopefully nicely into Ellie's talk next week on bioimage analysis pipelines. Okay, so I'm going to turn off my video so you don't have to look at me. Yeah, so we'll get going on sample preparation. Feel free to fill in questions in the Q&A and I'll try and answer them at the end. As in last week, we'll break for some questions. To give you guys the chance to answer some questions as we go through the workshop. So for each individual section, just to make sure you guys are following along with the content. Okay, so in terms of sample preparation, so yeah, we've got a couple of pages, as I say, on general rules. First thing that I wanted to highlight was that the best imaging will occur near the cover slip. Therefore, your sample should ideally be mounted on the cover slip and not on the slide. Because you're imaging through the cover slip, the best imaging is going to be at that cover slip interface. The further you go away from that, the more aberrations you're going to get as you go 3D into a sample. Ideally, most objectives are expecting a cover slip thickness of 170 microns. That is equivalent to a number 1.5 cover slip. If your lab has number 1 cover slips, then yeah, you can use those, but they're probably only okay for things like histological samples where you're only taking a 2D data acquisition, you're not taking 3D. The next thing is to select the best fluorophore for your experiments. And we covered a little bit of that in lecture 1 in terms of, the different parameters and things that we might consider with fluorophores. So does it match to the excitation wavelength on your equipment? What's the photo bleaching like? Things like that we start to look at. And that follows on to match the fluorophores to your microscope setup. So yeah, make sure that you've got the excitation and the detection wavelengths that are matched to the specific fluorophore that you're wanting to use in your experiment. One thing that we come across against with lots of new researchers is that they use a protocol that's been used in the lab before. I would always argue that you should probably go through and titrate concentrations of things specifically for your experiment. So if you're doing transfections, look at the DNA concentration, modify your fixative concentrations and ratios, especially if things aren't working. For your antibodies and stains, the companies will always tell you to use probably way more than you actually need to do. They have a vested interest in you buying more of the reagents. And one really key important thing is, especially if you're setting up an experiment from scratch, make sure to include positive and negative controls within that. That's a really vital experiment to do if you're doing a research thesis. Next rules we have are try and fix your sample as gently as possible. So you don't want to overfix and create artifacts within your sample. And you probably want to image your samples as closely to preparation as possible. So it doesn't need to be immediate, but certainly you don't want samples sitting around in the fridge for weeks or months before you come to image those samples. And last thing we're going to cover in this little section is the sample mountant. So please use the correct sample mountant for your microscope. This can come down to the immersion type. So essentially you want to match what your sample is mounted into the immersion type of the objective. So here we've got using an oil lens with a refractive index of 1.51. If our mountant is mounted in a media with a refractive index of 1.33, you can see we're not getting as nice image quality as we have matched that refractive index with our sample mountant. We're getting much nicer imaging in 3 dimensions than we do with the incorrect mounting media. The other thing to think of here is often we have researchers that buy mounting media with DAPI already included in the stain, in the mounting media. Normally, we would suggest you don't do that. DAPI has a really large emission spectrum, which goes into the green and often into the red wavelengths as well. This can cause really high background and start to deteriorate your image quality. So normally we'd suggest you use DAPI in solution, incubate it, wash it out, and then mount in a mountain without DAPI. Okay, so as I say, that's some very kind of high level general sample prep rules. Moving on from that, we need to start to think about what we're going to stain our samples with. So if we have cells or tissues, we've got a few decision trees here in terms of what we can use. So if we want to do live cell experiments, we're limited to either fluorescent proteins or dyes. Whereas if our samples are fixed, this also opens up the possibility of not just using fluorescent proteins and dyes, but also antibody labeling as well. And we covered that a little bit in the last presentation. With the dyes, it becomes really important that these are fixable dyes. So again, check the manufacturer's product information as to whether or not those dyes can be fixed. While we're discussing live cell imaging, that's another thing to take into account or to think about, is that you need to keep your cells alive throughout your imaging experiments. Sometimes we hear people say that, we tried to do a live cell experiment, but by the end, all of the media had disappeared. Obviously, your cells are not going to be happy well before the media has disappeared. So you need to think about how you're going to keep your samples as close to physiological conditions as possible. So things to think about are temperature. So in this picture here, we've got a microscope which is surrounded by this perspex box, which allows us to heat the microscope and the sample up to the correct temperature for mammalian cells. That's 37 degrees. We can control the gaseous environment. So often there is a stage top enclosure where we can control the CO2 concentration. That allows us to maintain the pH of the media, making sure that your samples are happy. One of the issues with the media disappearing is humidity. So if you pump dry air onto your sample, then you can have evaporation. So often we want any gas that's coming into the system to be humid, and you often get a bubbler type system like this within the flow of air that's coming in, just to make sure that that you can maintain humidity and stop that evaporation. The other thing we request researchers use is gas permeable seals, where you can actually seal in your sample. That still allows gas exchange, but doesn't allow evaporation. And certainly for longer term experiments, that can be very, very helpful. And then finally is making sure that there are enough nutrients in the media or enough media there to keep your samples alive and happy for an extended period of time. Next thing to think about is the microscope setup for live cell imaging. So for example, which microscope are we going to use? Do we have access to a spinning disk system? Do we have a confocal microscope with this resonance scanner, which scans the sample a lot faster and therefore is a lot more gentle? Some microscopes are equipped with this AreaScan, which allows either this fast mode or multiplex modes in AreaScan 2. Again, just scans the sample a lot faster and is therefore a lot more gentle. Or do you need to go to something like a lattice light sheet system, which is extremely gentle and very good way of doing, again, kind of long-term live cell experiments, or experiments where your samples are maybe prone to phototoxicity or photobleaching. We need to, again, select the correct fluorophores. So you want those to be as bright as possible. And therefore, you're trying to minimize the excitation levels required to image those samples. And we probably want to use the longer wavelength. So we don't want to be using ultraviolet light. So you probably want to avoid that Hurst or DAPI stain in the ultraviolet wavelengths. That's going to very quickly kill off your samples. Same thing for phototoxicity. So this is kind of intertwined. So longer wavelengths, avoiding ultraviolet light is preferred. And again, we want to minimize that excitation level that's going through. The other thing that we want to try and do, and that goes back to the kind of general guidelines, is we want to get the best image quality image through a cover slip or optical plastic. So sometimes we use these Sykes-Moore chambers. These are metal contraptions. So we've got a piece here which we can put our cover slip into. There's a silicon gasket which we can then kind of clamp down and put media in on top. Benefit of this is it's autoclavable. You can reuse it and sterilize it. Or you can use imaging dishes or slides like these. These are from iBiddy, Fluorodish. There's plenty of vendors that make these. But again, these are imaging through high quality cover slip thickness bases to get you the high quality image data at the end. So that's live cell imaging. What we'll look at next is sample fixation and labeling. So if you're trying to come in with antibody labels, often you are going to need to fix and permeabilize your samples to do that. There are a few common chemical cross-linkers that people use for their experiments. So things like glutaraldehyde, paraformaldehyde, acetone methanol. There's a relatively new one called glyoxyl. All of these have different pros and cons, and I've got them listed here on the slides. I won't necessarily go through them all, but certainly often like PFA and glutaraldehyde are used in combination to kind of make up for each other's strengths and weaknesses. But yeah, again, if you've never set up an experiment before, worth testing out a number of different ways of fixing your sample, because not all cellular compartments are fixed the same way with the same technique. Okay, so I mentioned why we would be fixing our samples, and that might be to do an IFA or immunofluorescence assay. And we covered the two types previously in the last talk. So primary, so direct and indirect. So the direct has a primary antibody conjugated with a fluorophore. Indirect, you need a primary and a secondary, which is then conjugated with the fluorophore. The general outline of these experiments is to either grow or attach your cells to a cover slip. If your cells are non-adherent, then you can attach them using something like a polyl lysine or a cell tack, which you treat on the cover slip surface to make the cells adhere. You would then come in and fix the cells. Again, as we mentioned, there are a number of different ways you can do that. And again, it's important to titrate those concentrations and conditions. To get antibodies into your cells of interest, you're going to need to permeabilize the cells. So a common tool here is Triton X100. Again, you can titrate the concentrations for best effect. And again, that's going to work differently for different organelles. Often, if you're trying to set this up from scratch, I'd be starting to look back in published papers, looking at similar structures to what you're interested in, to see what people have done previously. Once you've punched the holes in the outside of the cell, that will allow you to get your antibodies in and bind to your protein of interest. But in order to do that, you're probably going to want to block non-specific binding. And one of the things you can do here is either a bovine serum albumin, BSA, or use the serum from the secondary antibody species. So as I say, this helps reduce your non-specific antibody binding. Once you've blocked, you can go in and label, and then finally mount your samples as well. So that's for a kind of cell-based assay. What we'll do over the next couple of slides is to look at tissue sections and a couple of different approaches we can do there. One is paraffin embedding, and the other is cryopreservation. So let's start with paraffin embedding. So here you can isolate your tissue. We want to maintain the morphology and the structural antigenicity of the sample. Again, this is normally through some form of chemical fixation. And so formaldehyde-based fixation normally. We would embed that sample, so dehydrate initially and then embed into a paraffin block. We would then take a microtome as pictured here on the right-hand side and cut this into very thin sections. This is then put onto a slide. We can rehydrate. We go through antigen recovery either via heat or through enzymatic recovery, and then again go through that labeling step, so blocking an antibody labeling. We'll come through in a couple of slides' time and go through what the antibody labeling step would look like for most of these experiments. If you don't have any experience on tissue sections, then the University of Melbourne has a histology facility which can either train you to do that yourself or do that for you in a kind of fee-for-service type way. Similarly, for tissue experiments where we are cryopreserving the sample, so histology facility will do that too. This is a cryostat, which will section the cryopreserved samples. Here again, we're isolating the tissue. We're chemically fixing again, either in PFA or neutral buffered formalin. We're going to infiltrate with a sucrose solution before embedding into OCT. And we're freezing using the OCT. Sectioning on the cryostat, so this controls the temperature within the cutting. Again, we can dry the samples onto a slide. They can be stored long-term at -80 degrees if you need to come back and do the staining at a later date before you rehydrate those samples and then again go through the block and antibody labeling steps. So for each of those 3 examples I've just gone through, we haven't gone through the antibody labeling in detail. So that's the one thing I wanted to cover in this next slide. So we, when we're wanting to do our antibody labeling, and in this case, we're doing an indirect immunofluorescence assay. So we have our primary antibody here, which is detecting our protein of interest. And then we're using a secondary antibody, which has a fluorophore conjugated to it to detect that primary antibody. And again, it doesn't show here, but this allows us to bind multiple secondaries to this primary antibody, therefore amplifying the signal. So after we've got rehydrated the sample and blocked it to try and reduce non-specific binding, we'll go through and we will bind in the primary antibody. Really key thing for us to be aware of there is what animal that antibody was raised in. So was it raised in rabbit, for example? So that incubation can be normally, say, an hour or so. We would then go through and do multiple washes steps to remove any non-bound antibodies before coming back in with the secondary antibody label. And the key thing here is that this secondary antibody should match the species that the primary antibody was labeled in. So down here, we've got an example in this table. So our antigen is GFP we're trying to detect. We've got a mouse anti-GFP, so an antibody that detects GFP, which was raised in a mouse. Our secondary in this case needs to be an anti-mouse antibody, which was raised in goat and is conjugated to an Alexa Fluor 488. For the example of actin here, we've got an anti-actin antibody which was raised in rabbits, and our secondary in this case is an anti-rabbit, so detecting this part, which was raised in sheep and has an Alexa Fluor 568. So we just need to be careful of the species that our primary and secondary combinations have had. Again, once we've had probably about an hour's incubation on the secondary antibody, we would go through multiple wash steps to remove any unbound. You can optionally do a second fixation step to bind all of those antibodies together to the sample. Again, wash, and then finally mount the sample onto the slide and cover slip. Okay, so that's everything I wanted to cover on sample preparation. If you give me 2 seconds, I will try and find polls and quizzes so we can set that up. And I will launch that and give you guys a couple of minutes to go through and try and answer some of those questions, just to test your understanding of what we've covered so far in the lecture. Okay, so for question number 1, the question is, what is the best choice of cover glass for most confocal microscope applications? In this case, it is the one that 88% of you selected, which is number 1.5 cover slips or 170 micron thickness, which is good to see. The second question is, what should you take care when choosing an antibody panel? The correct answer here is that your secondary antibody should target the species that your primary antibody was raised in. So that's the second question there. And again, a large proportion of you guys got that answer correct, which is good to see. Okay, so let me just close that down. We'll go back to the presentation. Okay, so yeah, that was some kind of very high-level general guidelines on sample preparations and what to think about. Next, we're going to go into experimental applications, and we're going to spend quite a bit of time going through possibilities in this space. And there are a number of different techniques that are available. I've chosen to divide it up based on size of sample. So here we can go from kind of model organism side of things, so like fish and flies, whole bits of tissue from model organisms like mice, all the way down through tissues into cells. And then this red line here is actually what we call the diffraction limit. So this is at about 250 nanometers of wavelength. And this is for standard fluorescence microscopes, kind of the resolution limit. So because of the wavelength of light we're using, we're collecting in the kind of 500 nanometer ranges. Our resolution is limited to about half of that. So yeah, for standard microscopes can only see structures if they are greater than 250 nanometers apart. If we want to go below that, then we start moving into this green sphere area here, which is in the super resolution microscopy domain. Again, that will take us down to probably about, 10 to 20 nanometers. There's a few very specialist techniques which can go much, much lower than that now. Certainly we don't have those available within the platform. And yeah, if you need to start going down to atomic levels, then we need to start talking about electron microscopy, and that's not something we're going to cover in today's presentations. Okay, so we're going to start at the top end, at the kind of high sample side, and we're going to cover a few applications. So light sheet microscopy, multiphoton microscopy, slide scanning, high content imaging, and wide field microscopy. And we'll go through a few slides and give examples of how these work and examples of some of the data that's been generated. So light sheet microscopy is primarily used in developmental biology and neuroscience. It is a little limited in resolution in that we'll only give you cellular resolution. Again, there are lots of different flavors of light sheet microscopy. Here we're focusing primarily on large sample light sheet microscopy. As the name suggests, the sample is illuminated by a light sheet. So we have cylindrical lenses on this side and this side, which create what we call a Gaussian beam. Parallel to the beam, which is our excitation light, our sample is sitting here. Parallel to that is our detection arm. So we have an objective here and a camera going off into the distance. So here we're only illuminating our sample, which is in focus. So a single sheet of light going through in focus with the objective. We can then move the sample. In this case, it's showing a rotation within the sheet of light to build up a 3D data set. There are systems that will also translate the sample up and down through the beam. In this case, we're primarily using a dipping lens. So this little cuvette, as is drawn here, is often filled with some form of solution. And ideally, for this to work, the sample needs to be optically clear. So this is why it's primarily used in developmental biology. Things like Drosophila embryos, zebrafish embryos, and worms are fairly optically cleared. If you want to put larger pieces of tissue in there, then you need to chemically clear that tissue out, and that's something that we do at the university quite a bit. When it comes to tissue clearing, this has been around from a very, very long time, but can involve some fairly nasty chemicals. There are two main groups of methods that are used. So the first one is organic solvents. In this case, we're dehydrating the sample again, as we did for the tissue sections previously. And what we're trying to do is match the refractive index of the tissue with an organic solvent. So in this case, there's one approach here. This actually comes up under aqueous-based. This is the clarity approach. You can see a brain here before and after clearing. So you can see it's almost completely see-through. With the organic solvents, these are not always applicable to fluorescence proteins, so often the chemicals used will damage the fluorescent proteins and get rid of any fluorescence. Some that you may have heard of are things like BAB, disco, ethyl cinnamate is one. So a lot of our researchers use the disco-based techniques, and then finally image in ethyl cinnamate, which is slightly less toxic than some of the other chemicals. Some of the other clearing methods that are available are more aqueous based, so we can have removal of lipid. So systems like Clarity will do that, again followed by refractive index matching with higher refractive index solutions. So yeah, the approaches here are Clarity, Cubic, CDB, and Switch. The majority of researchers, as I say, at the University of Melbourne are using the disco-based techniques. Benefit is it makes the sample slightly smaller, and the sample is fairly hard and easy to manipulate onto and off the microscope. But yeah, the instrument itself can handle any of those techniques. One of the downsides here is it can be quite tricky and difficult to set up and quite expensive depending on which of the clearing approaches you decide to take. But again, we can help put you in touch with people that may have already cleared the tissue that you're interested in. One example data set that we have, this should be a video, but doesn't look like it wants to play. Let me just turn off my laser pointer. This is a guinea pig cochlea, which has been chemically cleared. That process takes about 3 weeks because this is primarily made up of bone, so decalcification is quite a long step. And here we can see the spiral within the cochlea. This group's actually looking at cochlear implants and looking at regions of damage caused by cochlear implants to see if they can improve the outcomes of those surgeries. All right, let's move on to multiphoton. So the light sheet in our example for large tissue is very much a fixed sample imaging approach. With multiphoton microscopy, we can actually do what we call intravital imaging on live anesthetized animals. And what we're doing here, if you remember the Jablonski diagram that we had from the last lecture, rather than having a single photon of light take us from the ground state up to the excited state, what we're actually looking is having, in this case, 2 photons hitting the molecule at the same time and causing the same effect. So in this case, we use longer wavelengths of light because they can penetrate into the tissue more deeply with fewer scattering effects, but we still get the same excitation as we would do for a single photon technique. So in this approach, we're pulsing photons onto the sample in the hope that, as I say, 2 photons will hit the molecule at the same time and cause this excitation effect. This means that to excite GFP, we'd normally be doing that with single photon at about 490 nanometers. With 2 photon excitation, we're using something more like 920 nanometers to get this 2 photon effect. And show you one example of this. This is one experiment looking at different T-cell immune cells within skin responding to a herpes infection. So in this case, we've got collagen in blue. In green is our herpes specific T cells. In red is our non-herpes specific T cells. And in magenta pink, we've got the herpes. And in this case, yeah, the group here from microbiology and immunology can quantify the motion of these T cells and see if there are different behaviors between the 2 different subsets. So very good for imaging, a few hundred microns up to potentially a mil into live samples, if that's what you're interested in. Next approach that we're going to look at is the slide scanner. This again is very much a fixed sample approach. So in this case, we would be taking either the paraffin or cryopreservation approach of fixing, embedding, and sectioning onto slides. The power on this system is that it is high throughput. So we can load in, this is our slide scanner, the AxioScan 7. We can load in 100 slides at a time onto the system, and we can get it to semi-automatically find the tissue and image whatever channels we want. We can do bright field and fluorescence imaging on here. We've got ranging from 5 up to 40x objectives on that system. And again, this is kind of meant to be plug and play. So you come in, set it up, press go, and then leave it to scan overnight. In this case, we have our preview scan showing where the tissues are. We can go in and define that. So we can use bright field or fluorescence for that. Here we have a few different examples of an overview scan, the same region at 20x and also at 40x as well. So again, it's not going to get you fantastic subcellular resolution, but if you're trying to count or visualize structures, then this is a much faster way of doing it than doing it manually on an epifluorescence scope or on a confocal microscope. Next thing we're going to look at is high content imaging. So similar to the slide scanner, this is high throughput and automated imaging. But in this case, we're not imaging samples on a slide or in a dish. We're actually going to mount samples into some kind of microtiter plate. So our system, which is the operator, can take anywhere from 6-well plates up to 384-well plates. And essentially, if you can stain your samples and specific regions of interest, we can set the system to automatically image each of these wells for you with multiple fields of views if you need. The real power on this system is the automated quantification. So you're going to generate thousands of images potentially from this plate. You don't have to be manually going through and counting each of those. So we spend quite a bit of time with the researchers going through and optimizing their quantitative workflows so that at the end of the day, you get a heat map or an Excel spreadsheet or a graph of what your data is rather than having manually to go through and do the analysis yourself. So the pros are automation, unbiased imaging, high throughput. We can do assay validation and screening. Lots of people here are doing like drug studies or multiple different treatments to try and look at combinations that work. Downside is it takes quite a bit of time to optimize. So sample prep is key. The image analysis workflow is key. It can generate huge amounts of data. And it's not specific for single regions of interest. We can't define this position in this well and this position in that well. It's just going to image the same region in each of the wells that you tell it to. Having said that, the possible applications are pretty endless. This is a few that we've done. So looking at migration assays, this is, I guess, a variation of the scratch assay that some people may have done, where there's a plug which you can remove and image over time and look for the movement of cells into the plug. We've done experiments over time where we can track cells, again, looking at kind of chemotaxis type experiments, or we can look at morphological analysis. So, what happens to cell shape after different treatments, gene knockouts, drug treatments, that type of thing. There's a really long, large list of things on the left-hand side there that you could do with these high-content screening systems. Next thing I wanted to cover was wide field. So we're going down in the scales and kind of getting more to the cellular level now. So we went through a wide field or epifluorescence microscope last week, and we touched a little bit on image deconvolution. So using a mathematical formula to reassign the out of focus light back to its starting point. This is really ideal for quite thin samples, so anything that's less than 20 microns. Things like cultured mammalian cells. The example shown here is a malaria parasite imaged in wide field mode in 3D, and then using that 3D information to deconvolve and create a kind of pseudo confocal quality image. As I mentioned, what we're doing is a bit of mathematics to restore back the fluorescence to its original starting point. We use what's called the point spread function of the microscope to do that. That's going to give us an increased resolution in all 3 dimensions, increased contrast, and also remove the noise as well. So you can see here, raw image and then deconvolved image on the right. What that requires is ideally Nyquist sampling. So we have these Huygens licenses, and if you're on a 60x objective with a 1.4 NA, then they ask that your pixel sizes are about 50 nanometers for this to work. So you want really good quality sampling. And also we need 3-dimensional information to assign that light back to its original focal plane. The Huygens licenses, which we have here at BOMP, have a wizard, which will take you through how to do this on wide field confocal area scan and light sheet data. That has both GPU acceleration to allow that to go faster, and also batch processing as well. So if you're doing the same thing over and over to multiple images, you can set it up and just leave it to run. This is, I guess, an overview of the process. So if this is what our light looks like, by the time it's gone through the microscope, it's a bit bigger and fuzzier just because of the way the optics have worked. What we're trying to do is use maths to bring it back to this original point. Okay, so that's everything I wanted to cover in your kind of up to cellular-based resolution. The next few applications that we'll go through will focus on cells, organelles, and proteins. And we're going to cover a few applications here again. So turf microscopy, confocal microscopy, a group of methods called the F experiments. And then we'll go into some of the super resolution methods as well. So confocal, super resolution, structured illumination microscopy, and then single molecule localization microscopy. So turf microscopy, which is also known as total internal reflection microscopy. So for standard wide field confocal imaging, we take so this example shows wide field. We're illuminating our entire field of view, our entire volume with the entire light coming from the entire objective. In turf microscopy, what we do is collimate the beam so it's a bit tighter, and we can translate it across to one side of the objective. What that allows us to do is to change the angle that light comes out of the objective. So for this wide field example, we have translation straight through. If we get the angle just right, then we can get this total reflection. So actually the majority of the fluorescence will bounce off the cover slip and reflect away. The benefit this has is that we're not illuminating the majority of our sample with the laser. What we get is something called an evanescence field, which will just illuminate the first 100 nanometers past our cover slip. Okay, so we're just seeing the structures within that first 100 nanometers into our sample, which in some ways limits the application of turf microscopy. But it can be very good for high contrast live cell imaging, and some super resolution techniques where we don't want to illuminate and potentially cause photobleaching or phototoxicity on our samples. We just want to illuminate this very thin optical section of the cover slip interface. And you can see an example here of an image taken in epifluorescence mode, not very much contrast, versus the turf mode, which is high contrast and also more gentle on our sample. So we have a couple of instruments available within our platform and a neighboring platform, which allow us to do that type of turf imaging. Okay, confocal microscopy is one of the kind of mainstays of our platform, and certainly a lot of biomedical imaging platforms. There are a lot of functionalities here that we could be getting into. Primarily, people are making use of automated stage functions, so the ability to take 3-dimensional data by moving the focus, moving the sample in relation to the objective and building up that kind of 3D model. A lot of researchers here at our university are using The automated stage also in the XY dimension to do kind of multi-point visiting experiments. So again, setting the system up to image multiple things automatically. You spend a bit of time at the start and it will acquire the data for you automatically. Or for capturing high resolution information over large regions. And one of the things I'll cover in the next slide is the tile scanning functions on confocals. But we can also use confocal for dynamic imaging, so things like live cell imaging, or the F experiments, which I'll come to in a few slides, which look at protein dynamics and protein-protein interactions. We can use the lasers to do photoconversion experiments. So we talked a little bit about photoconvertible fluorescent proteins in the last lecture, where we can specifically change the fluorescence emission wavelengths of certain molecules and track them over time. So we can look at protein and organelle dynamics. The other really important thing is, or the other possibilities are specialized detectors on here. So there are a few now super resolution microscopy specialist detectors, and we'll cover those in the super res section. But also the ability to do spectral detection, so across the visible light wavelength. So we no longer limited to just 3 or 4 channels. We can actually start to then multiplex our stains on top of them. Okay, so this is one example of the automated stage function. So here we have a single field of view which is taken at a lower magnification, which may be sufficient, but we can actually use a higher magnification lens to get good information. So this is an example here of a single field of view, which is imaged at this resolution. And we can use the stage functions on the microscope to actually take an acquisition here and move across, take another acquisition, move across. So we end up building up a high magnification, high quality image of a larger region, which gets stitched together into an individual image. And this certainly helps with, cell counting and that type of experiment. I mentioned about spectral imaging. So there are a few ways that can be done. So in this case, we have diffraction gratings, which separate out, again, the visible light spectrum. And we can move this position so that we're selecting just a specific wavelength onto our detector. Or there are also these detector arrays. So in this case, we're using a diffraction grating, spreading it out. In this case, this would probably be something like the Zeiss quasar system that has a 32 detector array. So we can actually take this in one shot, whereas this one, we would need to move that detection range across to take up a bit of time. That allows us to image and quantify intensity of each of our fluorophores across this visible light spectrum. So in this example, we have 9 different stains that we've been able to separate out. So one is autofluorescence plus 8 other stains, using single color controls to build up this spectra, and then a process called linear unmixing to try and separate out those individual channels. And this can be very powerful, but yeah, it needs a lot of controls in order to make sure that that's robust. We talked a little bit about live cell imaging. So one of the systems we have available at our platform is a spinning disk. So unlike a standard point scanning confocal where we have a single pinhole, the spinning disk system, as it suggests, has a disk which spins, and on the disk are hundreds of pinholes. This allows very, very fast acquisition. So this is detector on here is a camera, not a PMT. So the acquisition is much, much faster. And yeah, it can be more gentle than your kind of standard point scanning confocal, but often isn't as applicable to thicker samples. So what you might find is crosstalk between the pinholes here if your samples are quite thick and you get a lot of out of focus light coming from other focal planes. But yeah, it can be very good for live cell imaging. In this case, we've got a field of view with cells going through multiple rounds of mitosis, which often can be very prone to phototoxicity. If you're putting too much light on, you'll find that they'll hit a checkpoint and just stop dividing. So yeah, this is somewhere that you might want to use something like a spinning disk confocal. All right, I mentioned the F experiments, so there's lots of acronyms about to come your way for this. So the first one is FRAP, which is fluorescence recovery after photobleaching. The next one is FLIP, which is fluorescence loss in photobleaching. Then we have FFS, which is actually fluorescence fluctuation spectroscopy. And then finally FRET, which is Forster or fluorescence resonance energy transfer. And I'll go through over the next few slides what each of those are and when you might use them. So first one is FREP, so fluorescence recovery after photobleaching. In this case, we'd be tending to use a point scanning confocal, although you can do this on some other systems as well. Here we have a region which is fluorescently stained, and we have a specific region of interest that we're going to, as we're scanning across, increase the laser intensity to actually cause photobleaching within this region. So in image B, you can see that we've photobleached the fluorescence out of that region. What we're then doing is imaging over time and looking at the recovery of the fluorescence molecules from outside of that field of view into that region of interest. Okay, so we're looking at the mobility of the fluorescence molecules within this sample. So here we are up on A, pre-bleach. We then go in and bleach out the majority of the fluorescence. So this is step B. And then over time, we're looking at the recovery of the intensity within that region of interest. And there's a couple of quantitative readouts that we can use there. So we can look at the T-half, so how long it took to half recover, which gives you a readout of the mobility. But then we can also look at the immobile and mobile fraction. So how well did it recover back up into stability? Some of the applications here are things like membrane protein dynamics, although this is often relatively quick. Another good use that we've seen in the past is transport of proteins across the nuclear membrane, so through the nuclear pore complex, bleaching out fluorescence from within the nucleus, and then over time watching that recover back into the nucleus. So that was FRAP. A related method is FLIP, which is fluorescence loss in photobleaching. Again, we have a single region of interest here, but what we're actually doing is multiple bleaches within that region of interest over time. And what we're trying to look at is the continuity of the compartments, so from outside to inside the region of interest. So here we might measure the intensity on the membrane while we're bleaching in the middle of the cell. So we're trying to look at the connectivity between the membrane and the cell. Okay, so we're continually bleaching that spot, reducing the fluorescence in the cytoplasm. How does that affect the membrane staining? Is there a connectivity between those two compartments? The fluorescence fluctuation spectroscopy. So I only have one slide on here because this is probably a 1-hour lecture in itself. This is a group of methods that investigate mobility of molecules. It can provide information on concentration, diffusion coefficient, the number of molecules, and their oligomeric state within your sample. There are a few ways of doing that. So one approach is to illuminate just one part of the sample and look at molecules that come in and out of that excitation beam. This would give you this kind of jaggedy line here, which we can then fit and do different types of analysis on. So one common one is fluorescence correlation spectroscopy. This is pair correlation analysis. So looking at monomer versus oligomers. We can also do this on a confocal image, so using raster scanning as we normally do on confocal imaging. We can do things like numbers and brightness analysis, again, looking at oligomeric states, or an analysis called RIX. If you want to know more about that, then get in contact with us. We'll probably put you in contact with the labs at the university that do that on a day-to-day basis. Last F experiment I want to spend a bit of time on is fluorescence resonance energy transfer, also known as Forster resonance energy transfer. In this case, and again, I think we covered this a little bit in the first lecture, we are looking at two fluorescent proteins here, and what we're trying to look at is whether or not these two fluorescent proteins come into close proximity. So these are attached to, in this case, a sensory domain and a substrate. And what we want to ideally measure is when the substrate binds to the sensory domain. So when it doesn't bind, you can see here that we're exciting our CFP molecule, and that is emitting the light out from the CFP molecule at about 475 nanometers. When these two substrates and sensory domains come into place, this brings the YFP fluorescent protein into close proximity with the CFP molecule. And for FRET to occur, that needs to be within about 10 nanometers for that to work properly. Now, in this case, when we excite our CFP molecule, rather than emitting at 475 nanometers, it actually transfers the energy to the YFP molecule, which is excited at about those wavelengths. And in this case, we see emission coming from the YFP molecule rather than the CFP molecule. So we see a shift in the emission from 475 across to the 520 nanometers, 528 nanometers. So in this case, we can say that FRET has occurred. In order to set that up, what we ideally want to do is match the acceptor and the donor fluorophores. So in this case, your donor is the CFP molecule, so we're going to excite probably around here. What's that, about 430 nanometers? We've got our emission wavelength here. Ideally, what we want is a really strong overlap between the emission wavelength of your donor and the absorption or the excitation wavelength of your acceptor. Okay, so this green region here shows you that there's quite a strong overlap between the emission of CFP and the excitation spectra of the YFP. In terms of different applications that are possible, we can look at things like protein-protein interactions, as we showed before, things like calcium sensing. So when calcium binds to this reporter, it causes a structural rearrangement and brings these two molecules together, allowing FRET to occur. We can have the same thing with kinase activities. So if this protein domain here gets phosphorylated, then it causes, again, a structural rearrangement, allowing FRET. Or we can have the opposite, we have a protease activity which cleaves and results in no FRET happening. In terms of FRET analysis, there are a number of different ways that we can look at this. So something called acceptor photobleaching. This ideally is only performed in fixed samples. So again, with a bit of mathematics, we can work out the FRET that's going on here. We have ratiometric imaging. So this works where FRET pairs have fixed stoichiometries. So for example, the kinase or the calcium here, we always know we've got 1-to-1 for acceptor and donor in that model, in which case we can do a simple ratiometric imaging. If we do a method called sensitized emission, this requires a lot of controls to do mathematically. We would normally suggest if you want to do FRET, then we ideally would want to do that using something called fluorescence lifetime, which, if you remember from the first lecture, is the length of time that a molecule stays in that excited state. This characteristic is completely independent of the wavelengths of the molecules. So we'll spend a few slides going over fluorescence lifetime imaging microscopy. So as I say, some of the key experiments here are FRET-based experiments. So we can do what's called FLIM-FRET and really separate out the FRET really nicely using fluorescence lifetime. It also allows us to do multispectral analysis. So again, if we had dyes that potentially spectrally overlapped, so if we look over here in the table, we can see Alexafluor 488 and enhanced GFP. Well, they're spectrally very, very similar, but have very, very different fluorescence lifetime. So actually that allows us to separate out a sample with those two different stains on them. Can always also use it for autofluorescence imaging. So this is an example of a plant sample, unstained plant sample, which has been imaged using fluorescence lifetime. And you can see that, again, we can separate out specific parts of the tissue just based on their lifetime. What's happening with fluorescence lifetime imaging? Again, this is a pulse-based approach. So we're putting in pulses at specific time gaps, and we're measuring the time until the photons are emitted from those molecules. And we get a plot, something like this, which we can fit, and that comes up with our fluorescence lifetime. So again, a little bit more technically difficult than standard imaging, but yeah, certainly doable. This is one example of some of the analysis. So in this example, we've got a fixed stoichiometry, so 1-to-1 with our fret pairs. This is looking at histone compaction. And as we can see, as the molecules become more compact, the amount of fret increases. There's one way of looking at fret data, which is called a phasor plot, which separates it out onto this semicircle. And again, we can separate out these species within an image based on the changes of their fluorescence lifetime. Okay, so that's molecular imaging. What we'll do over the next few slides is go through some super resolution microscopy applications. And I've put together a table here which kind of identifies some of the more common ones. So as I mentioned at the start, so confocal microscopy, our resolution limit is probably in the order of about 250 nanometers in the axial direction, sorry, in the lateral direction, and about 600 nanometers in the actual direction. And yeah, ideally the best imaging is going to happen in the first 50 microns past the cover slip. One specialist detector, which is commonly used, which we'll talk about in the next slide, is this AreaScan detector. Also, the NSpark detector does the same thing from Nikon, or similar thing. Again, this is confocal based, so again, about 50 micron thickness of samples, unless you're going to clear them. This is giving us an increase up to about 100 nanometers resolution in XY and about 300 nanometers in the Z direction. We have stimulated emission depletion, so STED. That's possible to run in a couple of modes, so 3x, so 3D STED, or just standard STED. In 3D STED, we get even resolution of about 130 nanometers. Whereas in 2D STED, we can go down to about 40 nanometer resolution in XY, but that doesn't help our resolution in Z. We don't have a STED system at BOM, so we won't be covering that. The next approach is structured elimination microscopy. This is either a wide field or turf-based technique. So again, the best imaging is going to happen very close to the cover slip. This can go down to, companies are quoting down to 60 nanometer resolution with some fairly extreme post-processing, and will bring you down to about 150 nanometer resolution in Z. And then finally, single molecule localization microscopy. Again, this is done in either primarily turf, but also in wide field mode. Because it's turf, we're imaging very, very narrow ranges past the cover slip, but will give you the best resolution increase. So yeah, 20 nanometers standardly in XY. If you're not correcting anything in Z, then about 200, but yeah, we can also do 3D imaging, which will bring down the resolution to 50 nanometers in the third dimension. And the final system, which I won't cover today, is MinFlux, which is kind of a combination of STED and single molecule imaging, but I'm not aware of any of those systems within Australia. So we're going to start with confocal super resolution. So these slides are taken from Zeiss, but as I say, the Nikon NSpark uses a very similar approach. This is confocal-based technique, so we're point scanning across the sample. And it works on the understanding that if you narrow down the pinhole that sits in front of the detector on your confocal, then you can actually increase the resolution. So this is 1 AU, which is most people are standardly imaging at. If you narrow down the pinhole, then you can actually increase the effective resolution. So by about 0.2 area units, you're getting much better resolution than you would be doing at 1 area unit. The reason that people don't standardly do this is that by going to 0.2 of an area unit, you're blocking a large proportion of the light. So it's actually very difficult to get good quality signal to noise, good quality images to actually be able to do that and get the resolution increase. So what Zeiss and Nikon have done is a slightly different approach, where rather than having a single point detector detecting on the confocal, they have this kind of compound eye array of detectors. In this case, there's 32 individual detectors, and each has an equivalent area unit distance of 0.2 of an area unit. So without actually physically narrowing the pinhole, you're getting the kind of resolution increase. The clever bit of mathematics, then you can use that to increase the resolution on your image. In this example data here, same field of view taken on standard confocal imaging, and then also with the AriScan detector. And yeah, hopefully you can see there's a fairly good resolution improvement between the two data sets. One other thing that we've standardly done here is to use our Huygens license to actually deconvolve these AriScan data sets. So the top row here is an example of a cell which has been processed using Zeiss's AriScan. My phone's going off. That gives us about 120 nanometer resolution versus Huygens processing, which actually provides increase in resolution as well. So again, hopefully you can see visually the differences between those two. Next approach that I wanted to cover was 3D structured illumination. So this will give a twofold increase in resolution in both the X, Y, and Z dimensions, which gives you an effective 8-fold increase in resolution, allows you to do up to 4-color imaging. The downside here is you need to take 30 times more exposures than you would do on a standard confocal system. So, all right, let me just try and mute that. Yeah, sorry about that. So here we're illuminating our sample with a structured pattern, a known structured pattern. When this interacts with our sample, it causes these weird patterns here, which are called moire fringes, and they actually include higher resolution information. And again, with a bit of clever mathematics and physics, we can actually extract that higher resolution information out. So the images at the bottom here show a wide field of view with the same structured illumination image. This example shows a 3D sim that actually gives 120 nanometer resolution. This virus here is a virus called Hendra, which is about 300 nanometers across, if I remember rightly. The central image here again is a malaria parasite, again imaged in wide fields and in 3D sim applications as well. Next group of techniques that I wanted to cover was single molecule localization microscopy. So that again, there are a lot of acronyms here. There are 3 kind of commonly used approaches here. Essentially what we want to try and do is to generate this blinking pattern. So if I bring that video back up, this is actually the Eiffel Tower, which blinks at night, but it shows you how you build up a single molecule image. So we have our diffraction limited spots here. If they are sparse enough, then we can be sure that, say, this light here is coming from a single emission event. We can actually then fit that and place a kind of central point against that molecule. And then over time, we can build that up to produce a higher resolution image. Okay, so it takes a long time to build up single molecule data. And the real key is here is how do we make that blinking effect? So as I say, there are 3 kind of key possibilities here. First one is called PALM, photoactivated localization microscopy. Here we use specialist fluorescent proteins to allow us to generate this blinking effect. The one that we've used most is the storm approach, which is stochastic optical reconstruction microscopy. In this case, we're using synthetic dyes, so say antibody labeling, for example, with fluorophores attached. And we use reducing conditions plus strong laser intensity to push most of the fluorophores into a long-lived dark state, so they're not emitting. Only a few come back down and go through that fluorescence cycle, so they're actually emitting. And then the last approach is this DNA paint approach. Just put my laser pointer back on. And in this approach, what we're looking at is the binding of a DNA sequence. So you have an antibody which has a DNA sequence attached. Your antibody is detecting your protein of interest. We then come in with a complementary DNA sequence that's conjugated to a fluorophore. I'm looking at the binding of the two DNA sequences together on and off to cause that stochastic kind of blinking effect within the image. The nice thing here is that Gaussian fit that we're getting from the raw data can produce up to a 10-fold increase in resolution. So again, this is another malaria parasite imaged in wide field mode on the left, and then D-storm imaging on the right-hand side. Okay, so things gone a bit strange. We've gone through quite a lot of different applications here through the kind of fluorescence microscopy. So from large samples all the way down past the diffraction limit into super resolution microscopy. As I say, we won't touch on any of the electron microscopy applications, but if you have any interest there, then let me know and I can put you in contact with the facility manager of our electron microscopy core. Okay, so that was a lot of info to take in. What we're going to do is just quickly break. You guys can try and answer some questions again. Make sure I start the right one. Okay, so there's a couple of questions there. I'll give you again a couple of minutes to decompress and think about what the correct answers for those two questions might be. Okay, so for these two questions, the first question is, if you would like to identify subcellular structures which are greater than 250 nanometers, which is, if you remember, our diffraction limit, which of the following techniques would you choose? So in this case, the correct answer I had selected was confocal, but I guess you could also use slide scanner and D-storm, light sheet. The storm application would be primarily for structures which are smaller than that diffraction limit. So that's the super resolution technique. For question 2, which technique is often used for imaging full organisms or intact tissues which between 1 and 20 millimeters in size? In this case, ideally that would be the light sheet system for those larger samples. Certainly not storm, which, as I say, is for very small samples at high resolution. Okay, I think most people got those correct. So let's move on to the final part of the presentation. So we're going to go through a little bit about digital imaging and data management and image ethics. And yeah, as I say, hopefully this will transition you guys nicely into the talk next week from Ellie. So first thing when acquiring your data is to always save that as the raw file extension. So we've got a few there. And the reason we ask our researchers to do that is that it not only saves the image, but also the metadata as well. So it captures all of the information about how that data was acquired. So what laser, what objective, what filters, the detector, your pixel size, that type of thing. The other benefit here is on a lot of the microscope systems, you can use that file to actually open up and acquire the data again the exact same way. Okay, so you can copy that back into the acquisition software. Also, the metadata can be read into image analysis. So we know what the pixel sizes are, for example. Next thing, if you've got your raw file with your metadata, if you're going to export that for analysis, then we suggest that you use a TIFF file format. and that is because it is what's called lossless compression. So there's no compression going on. Your file size is large. Ideally, you should be using that for data analysis and publication. And I've shown a panel at the bottom here that if we take the same image as a TIFF file, convert it to a JPEG, and then subtract the two from each other, this is the information that you've lost just by doing that conversion. Okay, so if you do have data you've converted out to a JPEG, a GIF, or a PNG file, because of that lossy compression, you're losing information. Benefit is its file size is small. We would really only use that for presentation. So in a talk, printing into your lab book, into your thesis, that type of stuff. For publication, you're probably still better with the TIFF side of things. So definitely not for any data analysis. The other good habit to get into is to never process or modify the original file. If you are making any manipulations, then you should do that on a copy, and you should try and record every step of the modification process. So here we're exporting, we've got a copy here, we've done one thing to it, we've got another copy, we've done another thing to it, we've got another copy. So again, you can see the workflow that you've done on that data. Last thing is to think about data management. So it's a requirement of your funding bodies often that your data is kept for a certain period of time, and it should be doubly backed up. So you should have some form of disaster recovery. So always keep the original file safe and unchanged. Within our facility, we ask that you don't use the acquisition computer as a backup storage space. Try and get that off that computer as quickly as possible, because there are lots of other people that have access to that computer as well. Here at the university, you should have access to either cloud compute or research servers which have this disaster recovery. So if there's a fire in the data center, the university has a second copy elsewhere of all of your data. Next thing to have a think about is the fact that although we're creating an image, we're still creating scientific data. So essentially, it's an Excel spreadsheet with intensity values. We just see it as an image. So we have to be very careful about any modifications that we're making. So if we're trying to make the image more beautiful, we have to think of that as data manipulation. So what are we allowed to do and what shouldn't we be doing? And that's what I'm going to cover over the next few slides. So there are a number of resources available to you. There's a number of papers which I've included here, which give some guidelines on that. If you want to see real-life cases of image manipulation, then you can go to this Twitter page and follow along with real-life modifications. Okay, so what we're going to do is go through some of the examples, and I'll explain to you why these are good or not. In this case, we've got a Western blot, so it's not a microscope image. But here we've got a couple of modifications. So in this case, this band in the top here has been removed. In Photoshop, it's kind of scarily easy, too easy to do this. If you look down here, there's bands here which have actually been added back in. So they're making modifications to specific parts of that image. If you do want to, bring separate parts in from different Western blots, then yeah, they should be clearly marked and indicated as being from separate blocks. In this case, we've got a microscope image on the left-hand side. This is an electron microscope image. And in electron microscopy, you can label proteins of interest with antibodies. But rather than using fluorescence, you use nanogold particles attached to the fluorophore. So in this case, this sample has been stained with two different nanogold particles of different sizes. But the people here have decided that actually the staining isn't strong enough. I presume they're expecting the staining to be on these structures here. They've actually gone in and enhanced the contrast on those structures. So here we can see some larger nanogold molecules as well as some smaller nanogold molecules, identifying two different protein sets. The other thing they've done is gone in and modify this sample here. So the gold particles have been enhanced and the background dot has been removed, which again, hopefully you could see is fairly unethical. So they're making modifications to a specific feature on the image. In this example, our problem is our fold here in the tissue on the original image, which has been taken out in the modify image. Again, specific modification to a single feature within the image. There's a few things you could have done here, so we could leave it, especially if the fold isn't causing the change in the interpretation of the data. If it's not causing a change in the interpretation of the data, you could actually crop the fold out if the information you're interested in sits in this region. Or ideally, what you'd do is go back, resection the block, and take a new image that doesn't have a fold within it. In this case, we've got a fluorescence image, which looks reasonably good. That's the raw. What we can do is go and do a simple contrast adjustment here, and you can see these groups of cells here and over here have a very different background to the rest of the image. That suggests that they've probably been cut and pasted in from another image. So again, clearly unethical. You shouldn't be making these types of modifications within your images. So what modifications can we make? And yeah, to me, these are modifications we're making to the entire image. So in this example, we have an image taken here, which is, we haven't used much of the dynamic range. So this is our grayscale values here on the y-axis, so 0 to 255 grayscale values. So we've not used much of the dynamic range of the detector, which means our image is fairly dim. So for visualization purposes only, we could enhance the brightness of this image. Okay. So here it's making this a little bit brighter. We're stretching out the intensity values, so we've got a bit better signal to noise. Again, this is just for visualization, not for any kind of intensity quantification. However, if we go too far and restretch this too far, here we're actually hitting saturation. And now, yeah, we can't quantify the intensity of the values at these pixels across this line. So here it's saturated out. We don't know what the actual intensity values are. So we want to adjust it to increase visualization, but not too far that we're saturating the image. This is another modification to the whole image. So in this case, we've got a sample here with a GFP signal. We've got a positive control and a negative control, which to me is nice, right? You've got a positive and negative control within your experiment. Again, if we do a simple contrast adjustment here, then we can see that the negative control background is very different to the positive and the sample control background. So again, I guess this would raise questions as to how that data was acquired or has been handled. Was it acquired the same way, which ideally it should be if you're trying to compare within a single experiment, or has it been presented the same way? Yeah, so yeah, this just raises a whole bunch of questions about that experiment that probably could have been done a little bit better. Next thing is modification on the background. So here we've got a piece of tissue and they've taken out all of this background tissue. And they've also evened up the illumination as well. Here we're saying that this is unethical just due to the large degree of background alteration. Ideally, what you want to do with any modifications you're making to image is to clarify in the figure legend what's been done. It's then up to the reader to interpret, whether that was the correct thing to do or not, or I guess the editor as it comes to publication. Another one here is in the original image, there's a bit of a shading problem on the bottom right-hand corner here. This has been cleaned up. In this case, it would only be acceptable if no changes actually happened to the tissue. So it's just the background. And again, you've made a statement in the figure legend. To me, ideally, I would be saying that you spot that at the time of acquisition, you go away, realign the light path, and get an image that doesn't have that problem on it. And certainly if it's a bump microscope, please contact us if you see this type of stuff in your images. All right, so what is the best practice in terms of acquisition? And to me, it's to get the best representation of what you can see down the eyepiece in your final image, okay? So if you spend the time optimizing your sample prep, then the imaging side of it should be relatively easy. If you can't see your sample very strongly down the eyepiece, then it shouldn't be that the system, it's not a magic box. It can't suddenly create signal when it's not there. This is what I call the crap in, crap out rule, that if your sample is not very good, then your imaging shouldn't be very good. Just be careful of tweaking microscope settings to make samples look much better than they actually are. So some suggestions for best practice with image acquisition. You can amplify and suppress positive and negative signal. So your data should be calibrated to a known standard. So I prefer people to have a positive control within their experiments that they're normalizing samples to. Ideally, you want to acquire the data with identical settings between samples if you can. Identical post-processing steps if you can. Again, retain all original files for post-assessment and document each of those steps that you've gone through. Okay, so this is the final quiz that I'll force you to go through. Hopefully you like my generative AI pictures I've been using. Let me just go through and start off the last couple of questions just based on image ethics. Again, we'll give you a couple of minutes to get through that. Just share those results. Okay, so first question is, which of the below intensity histogram parameters is it ethically okay to change in an image for visualization purposes? I just realized I've actually removed that slide from the presentation. So well done to most people who got most of the correct answers there. So we can use the minimum intensity value on the histogram, which will clip out the kind of background. Ideally, the maximum intensity we can bring down again to rescale for visualization purposes. Those ideally are the two that we would use up until the point where we start to actually hit data, if you catch my meaning. I did also include gamma in there, although I'm not a huge fan of using gamma adjustments. But yeah, that is also possible. But again, you would have to say within the figure legend that that's what you've done. Which follows on to question number 2. Which of the following statements is correct? And the hint was select all that apply. So the correct answers here are modification should be performed to the whole image, and also modification should be reported in the figure legend. And yeah, it's good to see that most people got those answers correct. Okay. So that takes us to the end of the second lecture. So we've covered some kind of high-level general advice on sample preparation. We've gone through a lot of the different experimental applications that are possible. And then we've touched a little bit on image ethics and what to do and what not to do. And as I say, next week's lecture from Ellie on the introduction to bioimage analysis, we'll go through that in a little bit more detail.